Welcome back for part 2. Sorry guys, I had to stop and teach class. Life of a professor, right? So I'm silly behind. Okay, so anyway, now we get to talk about redox reactions. So much fun. So actually this is really cool. It's kind of like math and everything has to equal on both sides, and it's cool. It takes a little bit of practice to get it done, but redox reactions and balancing them is one of my favorite parts of chemistry. But then again, I'm a nerd and a dork and everything all of the above. So that's okay. Okay, so redox reactions. These occur in pairs, and there's an electron donor and electron acceptor, and these constitute a redox pair. Okay, the process salvages electrons and their energy. So, like one of the first laws of what was it, Newton or whatever, energy can neither be created nor destroyed. It's just transferred, right? So we're transferring those electrons, which electrons are energy, right? So this is just kind of showing you how everything's shuffling about. Just take a minute to appreciate how cool your body is that this is happening like all the time, right? And so anyway, so we're going to balance these electrons to energy, etc. And then the released energy can be captured to phosphorylate ADP or another compound. Okay, so phosphorylate ADP. Let's talk about ATP for a second here. ATP stands for adenosine triphosphate. The energy in ATP is stored in between the bonds of the phosphates. Okay, so when we break off a phosphate off of ATP, it becomes ADP or adenosine diphosphate, right? And then it releases energy. So phosphorylation is just the process of adding a phosphate on it. Okay, so if we take ADP and we add a phosphate to become ATP, it undergoes the process of phosphorylation. Side note. Okay, so anyway, looking at the slide, when we're talking about the oxidation, we're adding an oxygen to this molecule, and then it's reduced, so this would be the oxidative part of the reaction, and then the reduction part of the reaction. Okay. All right. Get back to the right buttons. Okay. So what about electron and proton carriers? Well, these repeatedly accept and release electrons and hydrogens to facilitate or help the transfer of the redox energy. And most carriers are going to be those helper coenzymes. This would be NAD, FAD, NADP, coenzyme A, and the compounds of the respiratory chain. And so you can see here, and like, we're going to talk a little bit about the electron transport chain, the citric acid cycle, etc. And so just remember that your inputs are going to be, I call them taxicabs, and I know that's kind of silly, but they're going to be empty taxicabs. So that's going to be like NAD plus empty. And then when it fills up with a hydrogen, then the output is going to be NADH. And so that's like basically what are they doing? They're just cofactors. They're helping carry their complexes, right? And so you can see here that NAD is on the left side. You have your adenosine and your ribose, okay? And then you have the oxidized nicotinamide, and then from the substrate you get 2 electrons. It goes on to here, and then so this is how NAD plus becomes NADH plus an extra hydrogen carrier. So again, they're shuttles, they're carriers, okay? And that's just showing you that one of the coenzymes is NAD plus, that becomes NADH. So then ATP, hey look, I got ahead of myself. So ATP is the currency of the cell, right? So it's money, 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 money. So it's the metabolic currency. There are 3 parts of the molecule, and this consists of an adenosine, which is a nitrogenous base, a ribose, which is a 5-carbon sugar, and then 3 phosphate groups, TP, triphosphate. And then removal of the terminal, or the outside phosphate, is going to release the energy. And the ATP utilization and replenishment is a consistent and constant cycle in active cells. Without ATP, guys, we would completely be dead. Okay? Period. We need to make ATP to be able to live. So you can see your adenosine backbone with the ribose. If you only have 1 phosphate, it's AMP, adenosine monophosphate. If you have 2 phosphates on it, it would be ADP, adenosine diphosphate. Then if you have 3, then that's going to be your adenosine triphosphate with the 3 phosphates on it. Okay, so that is that. I'm trying to see where this picture is in your book, but I don't think that this one came from your book. I think it was grabbed from elsewhere. But basically that just shows you what ATP is. Okay, So what about this phosphorylation of glucose by ATP? Well, this is figure 8.14, okay? And so, oh, that previous picture, I'm sorry, was 8.13, and that was the model of ATP, okay? So anyway, this one is the phosphorylation of glucose by ATP. And so this is, again, figure 8.14 in your book. So the first step in catabolizing or breaking down the glucose is the addition of a phosphate from ATP by an enzyme that's called hexokinase. Okay, so you can see here, glucose becomes glucose 6-phosphatate through this reaction, right? And then the use of this high energy phosphate is an activator and is a recurring feature of many metabolic pathways. So you can see that ATP broke off of one phosphate, it becomes ADP, and then that phosphate, because we can't create or destroy, we can just transfer, attaches onto that glucose, and it becomes glucose 6-phosphate. Okay, and this is through the reaction of the enzyme hexokinase. Remember, anything that ends in an ase is an enzyme. Okay. So, how do we form ATP? Well, this is figure 8.15 in your book, and ATP can be formed by 3 different mechanisms. The first one is substrate level phosphorylation. The second is oxidative phosphorylation, sometimes called OXPHOS. And then the other is photophosphorylation. Okay, we as humans can't do photophosphorylation because although we need sunlight to live, we can't use it to make energy. Okay, so the substrate level phosphorylation, this is a transfer of the phosphate group from a phosphorylated compound or substrate directly onto ADP. This happens in glycolysis, okay? And so it's just, I kind of teach this as a quick and dirty, right? It's not a huge process, but it gives us a couple ATP that when we need it, for instance, starting out glycolysis, right? Where oxidative phosphorylation, this is more like a sit-down dinner. It's more of a process. And this is a series of redox reactions that occurs during the respiratory pathway, specifically in the electron transport chain, okay? It requires oxygen and it produces a whole bunch of ATP. This is why the electron transport chain in aerobic metabolism creates our 30 ATP, right? And then that photophosphorylation, this again is ATP that's going to be formed utilizing the energy of sunlight, okay? So the substrate has been bound to the phosphate, creating a bond with high potential energy. And that's what you see here. And then the transfer of the high energy phosphate to ADP gives rise to an ATP and returns the substrate to its original form. Okay, so again, that's figure 8.15. So moving on and specifically talking about this, what is bioenergetics? And what are the pathways of bioenergetics? Well, bioenergetics is the study of the mechanisms of cellular energy release. And this includes catabolic, breaking down, and anabolic, building up reactions. And the primary catabolism or breakdown of fuels, glucose, sugar, right, proceeds through a series of 3 pathways. Okay, they're coupled, they are stacked. Okay, coupled is stacked, whatever. So step 1 is going to be glycolysis. Step 2 is the Krebs cycle. This is also called the citric acid cycle, CAC, right? And then step 3 would be the respiratory chain or the electron transport chain. And this is nicely summarized in figure 8.16 in your book. Okay. So looking specifically at the metabolic strategies, the nutrient processing is varied. Okay, and in many cases, it's based on 3 catabolic or breakdown pathways that convert glucose to carbon dioxide and also gives off energy. And those 3 pathways are going to be aerobic respiration, anaerobic respiration, and fermentation. And so aerobic respiration and metabolism, interchangeable. This includes glycolysis, the Krebs cycle, and the electron transport chain, which is the respiratory chain. Anaerobic respiration, on the other hand, does also include glycolysis and the Krebs cycle and the respiratory chain, but molecular oxygen is not the final electron acceptor. And then fermentation again starts with glycolysis, and this is where organic compounds are the final electron acceptors. Okay, so this is that figure that I was telling you about. This is figure 8.16, and this is a side-by-side comparison of the different stages and the major products of the pathways for aerobic, anaerobic, and fermentation. And note that not that all are going to begin with that process of glycolysis or breaking down of sugar, but they vary in much of the other pathways. and which ones are the electron acceptors and what the ATP yield is. So you can see that aerobic metabolism over here starts with glycolysis, produces an ATP as well as an NADH, and then the pyruvate get converted into acetyl-CoA. That acetyl-CoA goes into the Krebs cycle, and then that Krebs cycle is going to give off the FADH2 and NADH. Remember, those are our taxicabs, right, that are taking those hydrogens. Where are they taking those hydrogen electrons? Down to the electron transport chain. And then the important part of aerobic metabolism is that oxygen is going to be the final electron acceptor. In anaerobic, the processes are very, very similar. They're the same here, except for non-oxygen electron acceptors are going to be the final acceptor. So this would be SO4 minus, NO3 minus, or CO3 minus 2, okay? And so the maximum ATP that's going to be produced during aerobic respiration is 38, where in anaerobic is anywhere between 2 to 36. Where fermentation, this guy is kind of like the last ditch effort to kind of stay alive, but this is like in heavy exercises, right? So that's the only time that you would pretty much do the fermentation. But anyway, you have glucose. The first process is going to be glycolysis, and again, you're going to be making those 2-pyruvate. Then the 2-pyruvate kind of say, crap, I don't have oxygen, kind of want to stay alive, so what am I going to do? Well, you undergo this fermentation, and what fermentation does is it actually regenerates a NAD+, so that we have an empty taxicab, so it can go back up and do glycolysis and limp along, right? We can at least have a couple ATP. But in the process of doing that, we make lactic acid, and so we actually have to have another process to take care of that lactic acid. But regardless, the organic molecule is the final electron acceptor, and the maximum ATP produced is 2. And actually, taking care of this lactic acid takes up more energy than that, so we end up losing it. But regardless, in heavy strenuous exercise, this keeps us alive, and we have ATP at the ready. So that's always good. So specifically looking at aerobic metabolism, and again, this is the close-up of that figure 8.16. Aerobic metabolism is going to be a series or enzyme catalyzed reaction in which electrons are going to be transferred from fuel molecules, glucose, sugar, my favorite food group, to oxygen as the final electron acceptor. We have glycolysis, which I call step 1, and this is where a 6-carbon glucose is oxidized and split into 2 molecules of pyruvate, otherwise known as pyruvic acid. And again, remember, things have to equal. So if we have 6 carbons and we split it into 2, that means that our pyruvate are each going to have 3 carbons, right? And then NADH, our cofactor, is going to be generated. So we have 1 full taxicab. The citric acid cycle is also known as the Krebs cycle, and you can see that that's labeled here. So citric acid cycle, Krebs cycle, and there's another name for it too. The other people call it. There's like 3 names for it. I have a big metabolic process behind me, so that's why I look behind me to look. Anyway, this is going to take that pyruvic acid after it's been turned into acetyl-CoA, and it's going to process it further. And during doing this, we're going to be producing CO2. So fun fact, when you exhale, and we exhale carbon dioxide, this is where we're getting it from, okay? We're going to generate 3 CO2 molecules. We also have our full taxicabs, NADH and FADH2, that are going to be generated. And then the final step of aerobic metabolism is going to be the electron transport chain. And this is where we're going to accept electrons from NADH and the FADH2. Okay, so our full taxicabs. And then we generate energy through a sequential redox reaction, which is called oxidative phosphorylation. Okay, so you can see kind of the details of all of these guys right here. Okay. So what is glycolysis? Well, glycolysis is the breakdown of sugar, okay? And so it is a complicated little process that's actually kind of cool. The enzymes are named for what they do, and so once you figure that out, it's kind of like straightforward. So figure 8.17 in your book. This is the summary and the steps of glycolysis. And so the primary reaction steps are described in the textbook, okay? And these reactions all take place in the cytoplasm, or the cellular glue is what I call it. And this happens in the cellular glue or the cytoplasm, regardless of the cell type. So this is the first part of aerobic metabolism, okay? So that's what this is showing. that it's going to be the first part of aerobic metabolism. So all of this right here is glycolysis, and glycolysis is just this kind of orangey box here. And let's see, it says only the pink carbon skeleton is shown to simplify the dynamics of the reaction. Okay, so it's like has the 3 carbons here, 3 carbons, and so it's not showing the whole molecule, it's just showing your carbon backbone, just so that it doesn't get overwhelming. And then a detailed analysis of the specific chemical structures and reactions can be found in Appendix A if you want even more information. But basically, it's really cool. You take the sugar, you break it down through all these enzymatic reactions, and you end up getting 2 3-carbon pyruvates. So then what happens to pyruvate? Well, this is in figure 8.18, and this is the fates of pyruvate. So this metabolite, or pyruvate, is in a very important hub, and it really is, okay? And it helps to decide, hey, what are we going to do? And so it's a very important hub in the processing of nutrients by microbes. It may be fermented anaerobically to several end products, or it can be oxidized completely through aerobic metabolism that we just talked about, to carbon dioxide and water through the Krebs cycle, otherwise known as the citric acid cycle, and the electron transport chain. And it can also serve as a source of raw material for synthesizing amino acids and carbohydrates. And that is detailed in figure 18.25, okay, which we'll talk about later. So, but basically, glycolysis produces this pyruvate, and then the pyruvate can be stored as extra, like if we have extra pyruvate, we can store it as amino acids, as sugars, or as fat metabolites. Or it can be transferred into acetyl-CoA, which then goes into the Krebs cycle and then down into the electron transport chain through the cellular respiration cycle. Or it can undergo anaerobic through the fermentation process, and it can be turned into lactic acid, lactate, right, and gases, or an aldehyde, and be converted into alcohol, acetone, or 2,3-butanediol. So it's very important. So this is the Krebs cycle, and this is found in process 8.19, or in picture. I have a wobbly chair. If you guys wonder why I keep standing up and stuff, it's because I wobble and then I have to readjust. Okay, so anyway, the reactions of a single turn of the Krebs cycle. And this is this middle highlighted area in the all over aerobic metabolism picture. So this one here is the Krebs cycle, which is also called the citric acid cycle. So you can see over here, okay, this is step 1 all the way down. So in step 1, the 2-carbon acetyl-CoA molecule combines with oxaloacetate, and this forms a 6-carbon citric acid, citrate, and it releases the coenzyme A, okay? So this is step 1 that's here. And then the citrate in step 2, which is going to be right here. So the citrate changes from the arrangement of atoms to form isocitrate. And then step 3, which is down here, isocitrate is converted into a 5-carbon alpha-ketoaldehyde butyrate, which yields NADH and CO2. And then down here in step 4, the alpha-ketobutyrate reacts with coenzyme A, and it loses the second CO2, and it generates another NADH plus a 4-carbon succinyl-CoA. And then down here in step 5, the succinyl-CoA is converted into succinate, and it regenerates that the coenzyme A, and this substrate level phosphorylation is going to produce an ATP. And then in 6, which is over here, the succinate loses 2 hydrogens and 2 electrons, yielding fumarate and generating FADH2. And then in step 7, which is over here, the fumarate reacts with water to form malate. And then in step 8 over here, an additional NADH is formed when malate is converted to oxaloacetate, which is the final product. And then this is ready to re-enter the cycle and react with another acetyl-CoA. So then you can see it keeps cycling. And this it can happen like one at a time. So it's kind of like a slower process, but it needs to keep going. So that would be the overall of the Krebs cycle. So then moving on to the final big boy, then this is the electron transport and OXPHOS, or oxidative phosphorylation. So the final processing of electrons and hydrogens and the major generator of ATP happens in the electron transport chain. And so that chain of redox carriers that receives electrons from the reduced carriers, again, those are NADH and FADH2, the electron transport chain shuttles the electrons down the chain, and energy is released and subsequently captured and used by ATP synthase. And remember, synthesis is a fancy word too. for to make. So what does it make? ATP. Okay, so like I said, enzymes are a lot of times named for their function. Okay, so the ATP synthase complex produces ATP, and this is through the process of oxidative phosphorylation. Okay, so this picture is electron transport and chemiosmosis. So this one is going to be very similar to the one that's in figure 8.20. Slightly different, but basically the proton transport chain by the electron transport system into the inner membrane space, and it sets up a chemical and charge gradient between the inner membrane space and the matrix, and then this is called the protein motive force. And then we have our ATP synthase, and that's over here. So basically what these little guys are, these are complexes, and this is where they're dropping off those hydrogens. So your input's going to be NADH, your output's going to be NAD+, because you just drop off those hydrogen passengers, okay? Then those hydrogens are shuttled across the membrane, and they go against their concentration gradient. They get really high up here, and then they're going to flow down the ATP synthase to make our ATP. So basically the gradient creates an electrical potential for capturing the free energy of the electron movement to drive the ATP synthesis. And as protons, or those H+, diffuse from the inner membrane space into the matrix, the F1 portion rotates and pulls in ADP and a PI, an organic phosphate, so that's down here, okay, and it releases the ATP. This is an exergenic reaction that requires 3 protons for each ATP. Okay, so again, this is kind of the close-up of what we just talked about, and you can see that, again, those electrons that were grabbed from the Krebs and glycolysis are shuttled across through the complexes, right? And then they become higher concentrated here, and then they get driven down through the ATP synthase to make ATP. And then another thing is the final electron acceptor here is going to be oxygen. So this is an aerobic process that requires oxygen so that we can form water. So that's kind of one of the reasons when you go like that, you breathe into your hands, you have moisture. One of the reasons. There are multiple, okay? So then what about the formation of ATP and chemiosmosis? Well, what is chemiosmosis? As the electron transport carriers shuttle electrons, they actively pump hydrogen ions, or protons, across that membrane, setting up the gradient of hydrogen ions. This is the proton motive force. This is what we just talked about in the previous slide. Then the hydrogen ions diffuse back through the ATP synthase complex, causing it to rotate, and this causes a 3-dimensional change that results in the production of ATP. So this would be like a close-up of it, and this is figure 8.21 in your book. And so as the carriers in the mitochondrial cristae, or those little folds, transport the electrons, they're also actively pumping those hydrogen ions, or those protons, into that inner membrane space, producing a chemical and charge gradient between the outer and the inner membrane space. Okay, and then the distribution of those electric potentials and the concentration gradient of protons across the membranes is going to drive the synthesis, or the making of ATP, by that ATP synthase. Okay, and then the rotation of this enzyme couples the diffusion of hydrogens into the inner complex compartment with the bonding of ADP and of phosphate. And then the final event of the electron transport chain is the reaction of the electrons with the hydrogens and the oxygens to form metabolic water, H2O. And this last step is catalyzed or helped by the cytochrome oxidase, which is the cytochrome AA3. Okay, so it has another complex to help it out. All right, so this specifically is figure 8.21, and this is the chemiosmosis in bacteria. And so this is an enlarged view of the bacterial cell envelope to show the relationship of the electron transport chain, where it happens, and ATP synthesis. Okay, so bacteria have the ETS, the electron transport system, carriers in the ATP synthase that are stationed, or they live in the cell membrane. So they're just, as you can see, it goes across the cell membrane. As the ETS shuttles the electrons that are delivered by our taxicabs, NADH and FADH2, some of the ETS carriers also transport protons into the periplasmic space between the membrane and the cell wall. And this sets up a gradient that's very similar to what happens in the mitochondria. The bacteria are going to vary in the exact nature of the carriers of the electron transport system. So what is the terminal step? Terminal meaning final. Okay, well, the oxygen is going to accept 2 electrons from the ETS, and then it picks up 2 hydrogen ions from the solution to form a molecule of water. So oxygen, like I pointed out before 2 slides ago, is the final electron acceptor. Okay, so that's why this is an aerobic process. So the theoretical yield then for our ATP, for aerobic metabolism, is going to be, we have 6 ATPs, 2 ATPs, 6 ATPs that come from here, 18 ATPs, 4 ATPs, 2 ATPs. And so the books vary. So like in my other class that I teach, we teach it as 30. This one teaches it as 38. Some of them see 32, some of them are 36. For this book, for this test, memorize 38. But the reason why we don't have a concrete answer is because, you know, the body is very dynamic and there are a lot of different ways to calculate things. But I mean, like I said, nothing, like we haven't quite found a concrete number. But again, for this class, we are going to end up using the word or the 38. Okay. All right. So then moving on to anaerobic respiration. So this functions like aerobic respiration, except for it uses or utilizes oxygen containing ions rather than free oxygen as the final electron acceptor. So this would be nitrate, which is NO3 minus, or nitrite, which is NO2 minus. And most obligate anaerobes use hydrogens that are generated during the glycolysis and the Krebs cycle to reduce some compound other than oxygen. So what is fermentation? Well, fermentation is the incomplete oxidation of glucose or other carbohydrates in the absence of oxygen. So if you've ever made beer, wine, anything like that, right? We know that fermented foods, you need it to have no oxygen. So you put it in a closed container so that the oxygen can't get to it. So this is in the absence of oxygen. And this uses organic compounds as the terminal electron acceptors. And this is only going to yield a very small amount of ATP. The production of ethyl alcohol by yeast acting on glucose is one example of fermentation. We also can form an acid, a gas, and other products by the action of various bacteria on the pyruvic acid. And so this would be, again, the snippet of fermentation that was in that figure on your book. Okay. And so that comparative figure, which was very nice, and that was, again, in figure 8.16. So looking specifically at alcoholic and acidic fermentation, well, we have this very fancy figure, which is going to be figure 8.23 in your book. So these are the fermentation systems that produce acid and alcohol. So in both of these cases, the one on the left and the one on the right, the final electron acceptor is an organic compound. In yeast, pyruvic acid is decarboxylated to acetaldehyde and CO2. And the NADH that's given off in the glycolytic pathway reduces acetaldehyde into ethyl alcohol. In homeolactic fermentative bacteria, the pyruvic acid is going to be reduced by NADH to lactic acid. And both systems are going to generate NAD+, which then can feed back into glycolysis, like I said before, and keep glycolysis limping along. Or other citric or other cycles, such as like the pink dotted lines. Okay, so basically the purpose of fermentation is going to be regenerating the NAD plus so that it can go back in and at least do the glycolysis pathway. Okay, so what are the products then of our pyruvate fermentation? Well, this is figure 8.24 in your book. And these are miscellaneous products of pyruvate fermentation and some of the bacteria that are involved in their production. So you have mixed acids, right? You have pyruvate can turn into ethanol, formic acid, lactic acid, acetyl-CoA, oxalic acid, acetoacetic acid, so a lot of different things. So pyruvate is incredibly versatile and we really need it, okay? So when we want to compare and contrast aerobic respiration and fermentation and anaerobic respiration, then we have this handy-dandy little table that was shown at the very beginning of this chapter. Okay, so you can see that you have pathways that are involved, etc. So in my book, it's page 238. So you have the pathways that are involved, the final electron acceptor, the products, and then the primary pathway that's found in. So this would be a great one to have a flashcard or to be able to draw a picture so that you can really understand it. So what about the biosynthesis and the crossing pathways of metabolism? So many pathways of metabolism are bidirectional, or otherwise known as amphiphobic. Okay, so they can go in multiple different directions. And so this is figure 8.25 in your book. So an intermediate compound such as pyruvic acid and acetyl coenzyme A serve multiple functions. With comparatively small modifications, these compounds can be converted into other compounds and enter a completely different pathway. Note that the catabolism of glucose, which is in the center, right? So that would be there, or here, right there. It furnishes numerous intermediates for anabolic pathways that synthesize amino acids, fats, nucleic acids, and carbohydrates. And these building blocks can serve in further syntheses of larger molecules to construct a wide array of cell components. So you can see things are multi-purpose. Our body is very efficient at using things that we have and using them for more than one function. Okay, so again, you can see that this is the figure a little closer up, and again, this is figure 8.25 in your book. You have your cell structure, the larger molecule, the smaller constituent of that macromolecule, and then the pathways that they can be involved in, and then the simple products that can be produced. So what about this biosynthesis and the crossing of the pathways in metabolism? Well, the catabolic or braking pathways contain molecular intermediates, otherwise known as metabolites, that can be diverted into those anabolic pathways too. Pyruvic acid can be converted into amino acids through amination. Amino acids can be converted into energy sources through deamination. And then glyceraldehyde 3-phosphate can be converted into precursors for amino acids, carbohydrates, and fats. So you can see we're multifaceted, our bodies. So what about this and reactions that produce and convert amino acids? Well, this is figure 8.26 in your book. All of the reactions require energy as ATP or NADH and specialized enzyme. In amination up here, through amination, which is the addition of an ammonium molecule or an amino group, a carbohydrate can be converted into an amino acid. In B here, in transamination, which is the transfer of an amino acid group from an amino acid to a carbohydrate, the metabolic intermediates can be converted into amino acids that are low in supply. Where in C, in deamination, which is the removal of an amino group, the amino acid can be converted to a useful intermediate of carbohydrate catabolism. And so this is how proteins are used to derive energy. So ammonium is the waste product of deamination, and note that carbohydrate structures are in brown and amino acids are in blue. Okay, so very, very useful, very, very beneficial. Okay, so moving on to section 8.6 in your book. What about photosynthesis? We as humans can't do it, but a lot of microbes can. Okay, so this is the Earth's lifeline. So the ultimate source of all of the chemical energies in the cells come from the sun. So we have 6 CO2s plus 6 waters, okay, plus light and pigment is going to end up giving you C6H12O6, which is the chemical formula for glucose, and 6 oxygens. So what is photosynthesis? Well, this is figure 8.27 in your book, and it occurs in 2 stages. We have light dependent, which is where the photons are going to be absorbed by chlorophyll, carotenoids, and the phytocoagulin pigments. And this is when water is split by photolysis, releasing the oxygen gas, and that provides the electrons to drive the photophosphorylation. This released light energy is going to be used to synthesize ATP as well as NADH. The light dependent photosynthesis, can occur in 2 stages: light dependent and light independent. And so the light independent reactions are going to be dark reactions, and this is called the Krebs cycle. And this uses ATP to fix carbon dioxide to ribulose 1,5-bisphosphatate and convert it into glucose. Okay, so this is, we exhale carbon, this is how it gets back into like the oxygen cycle, okay, is photosynthesized. So looking specifically at these photosynthetic reactions, this is very similar to what is shown in figure 8.23. So basically, you can see here the cell of the motile algae, okay, Chlamydomonas, with a single large chloroplast. The chloroplast contains membranous components where chlorophyll molecules are the photosystems for the light reactions are located. Okay, you have a chlorophyll molecule with its central magnesium and atom that's held by a porphyrin ring. And then C down here are going to be the main events of the light reactions shown in an expanded view, okay, with one granium. So when the light activates over here in photosystem 3, it sets up a chain reaction in which electrons are released from chlorophyll. Then these electrons are going to be transported along the chain carriers to photosystem 1, kind of like in the ETC, right? And then down in 3, where did we go? 3 down here. Okay, the empty position in photosystem 2 is going to be replenished by the photolysis of water, and then the products of photolysis are oxygen and hydrogen. And then down in 4 over here, the pumping of hydrogen into the interior of the granium produces conditions for ATP to be synthesized. And then in 5, the final electron and hydrogen acceptor is going to be NADP, which receives these from photosystem 1. And then here in 6, both NADPH and ATP are going to be fed into the stroma for the Kelvin cycle. Okay, so some similarities actually, okay? And then what are these Kelvin cycle reactions that they are talking about? Well, you can see here, let's see if I can find it in your book. I'm going to pause first. So the similar figure in your book is going to be figure 8.29. And so this is the Kelvin cycle. And so the Kelvin cycle includes the main events of photosynthesis during which carbon is fixed into the organic form using the energy, which would be ATP or NADPH. And this is going to be released by the light reactions. The end product, which is glucose, is going to be able to be stored as complex carbs or sugars, right? Or it can be used in various amphiphobic pathways, so they can be used for more than one function, right? To produce other carbohydrate intermediates or amino acids. Some pGal is going to be used in the regeneration of RuBP, which uses additional ATP made during the light-dependent reactions. Okay. And that is all. So thanks for coming to this lecture, and we will see y'all in class. Thanks. Bye-bye.